I'm Daniel Levine, and this is the Bio Report. Despite the emergence of new modalities and drug development technologies, the cost and time to produce new therapies has changed little, and failure rates remain high. Xaira aims to change that with a systemic, AI-driven approach that tackles three pervasive bottlenecks: choosing the right targets, designing the right molecules, and matching the right patients, by running as much work as possible in silico and using high-dimensional causal datasets to train virtual cell foundation models. The company is initially focusing on high-value, historically undruggable targets and ultimately on building a pipeline of differentiated biologics. We spoke to Marc Tessier Lavigne, co-founder and CEO of Xaira, about applying end-to-end AI across target discovery, molecular design, and patient stratification, the company's more than $1 billion in funding, and how it seeks to enable a new generation of scientists fluent in both AI and biology. Marc, thanks for joining us.
Well, thank you, Danny. It's great to be here.
We're going to talk about Xaira, its vision of itself as a new generation drug company, and its efforts to remake pharmaceutical R&D with an end-to-end AI approach. Let's start with the problem you're seeking to address. What's wrong with the traditional approach to drug discovery and development?
Well, we made great strides over the past several decades with drug discovery and development, especially in opening up new fields, new modalities. Think about bioconjugates, armed antibodies. Think about RNA vaccines and the like. But at the same time, the overall productivity of the sector has not improved much. If you go back 20 years, the figures that were discussed was that on average, from starting to work on a target to making a drug and then pushing it through clinical trials to FDA approval would take on average about 13 years. Attrition in the clinic, drugs entering the clinic, 90% to 95% would fail. And the cost was in the billions of dollars. Dial forward 20 years and the numbers pretty much haven't budged. So although there have been great successes over the past several decades, especially in opening up new fields, at the same time, as a whole, we know, first of all, there are opportunities that we're not able to access. And secondly, we just have to do a better job of fixing the overall process, reducing the timelines, increasing success rates, reducing attrition, and thereby lowering costs. The way we do business today just isn't sustainable in the long run. And so there's a need to rethink this whole process of drug discovery and development.
What's the potential for an integrated AI approach to fix that? And is it fixing it by addressing the high failure rate, or are there other ways that it's going to reduce cost and compress timelines?
Well, I think you have to use the word integrated approach. And I don't know if this is integrated or not, but maybe the word I would use is systematic AI approach, applying AI to all the different steps of drug discovery. There are some areas where there's already been huge progress just in the past few years, especially since the advent of ChatGPT, large language models, and the like. If you think about logistics and operations, people have been deploying these types of approaches to deal with your regulatory documents and filings with the FDA, to identifying sites for recruiting patients, to enrolling patients and the like. So we're already seeing a lot of progress in terms of logistics and operations, again, with time savings and cost savings. The bigger prize will be to tackle the scientific dimensions of the drug discovery process. To make a drug, I think we all know you have to do three things. You first have to choose the molecular target that you want to modify, typically inactivate, but sometimes activate with your drug. Then you have to make a drug that has the desired characteristics of activating or inactivating the target and with the appropriate specificity and reaching the appropriate tissues and so forth. And then you might have the best drug against the best target. If you develop it in the wrong patients, it will fail. In fact, a lot of the failures in phase 2 are because we have a good drug and a good target, but we can't figure out who the patients are. So this problem of identifying the patients who are most likely to respond, of stratifying the patients, is the third step. Each of those steps, identifying targets, making the drugs, and stratifying the patients, is currently very inefficient. There's a lot of science that's involved, but there's a lot of empiricism, trial and error, and a lot of failures. And it's that we have to fix if we really want to make a big difference. And that's where we believe that AI can make a difference to all three steps, to choosing targets, to making the drugs, the therapeutics, and then to matching targets to patients. That is what will really move the needle when it comes to timelines and success rates and cost.
Xaira talks about turning an artisanal process into an engineering discipline. I've heard synthetic biologists use this language for more than 20 years. What does Xaira mean by that? And given that the computational core of the company is built on the work of Xaira co-founder and Nobel laureate David Baker and his team at the Institute of Protein Design at the University of Washington, do you think of what Xaira is doing as using synthetic biology to produce therapies?
Well, I'll address your second question later, but maybe we can go back to the first one about what do we mean when we say turning an artisanal process into an engineering discipline. So what I described, as those three steps and how we go about target ID, drug making, and patient stratification, is artisanal. Again, a mixture of science, but also a lot of art and experience and trial and error. When we say we want to turn it into an engineering discipline, what we mean is we want to be able to do much more of the work in silico and less in the wet lab or in the clinic, and with higher success rates. And an analogy might be helpful here, if you'll permit me. Think about the field of aeronautics, and if you go back, a century, century and a half, early 1900s. If someone wanted to design a new airfoil for an airplane, we had equations for fluid flow and airflow that could be solved, difficult equations, and without supercomputers. They were often done with approximations. So what would happen is that the designers would design things up to a point, but very rapidly you'd go into the wind tunnel, you'd make a prototype, go to the wind tunnel, test it, and adjust, go back to the equations, and go through multiple cycles of improving the aerofoil bit by bit. Supercomputers come along, and suddenly the calculations are much more precise. That doesn't negate the need to go into the wind tunnel, but what it means is you don't have to go as often. Very rapidly, you get much closer to the finished product. You can go into the wind tunnel, do some minor tweaks, and voila, you have your aerofoil. So that's the going from artisanal to engineering in that case. So how do we port that analogy to biology and to what we do for target ID or patient stratification? Well, here we have no equations in biology. People try to derive equations for the behavior of cells, what's happening at a molecular level inside cells. This was actually quite a big area of focus in the 70s, the 80s, the 90s, even to the early 2000s. But basically, this whole program was unsuccessful. And the reason is biology is much too bespoke. It arose through evolution, through the accretion of all these different parts that somehow miraculously function together. So we don't have equations for biology, and we won't have equations for biology at any time soon, if ever. So how do we go about modeling biology? Well, there, AI is, I believe, and I think many believe, the correct tool for modeling biology. Eric Schmidt has this wonderful saying, which I believe is absolutely true and I believe very profound. which is that AI is to biology what math is to physics. So in the case of physics, since Newton, we've known that we can model the physical, the inert universe using mathematics. In the case of biology, we cannot. But AI, in fact, because of the ability of AI, if you feed it with the right kinds of data in the right quantities to discern patterns, the ability of AI to discern those patterns enables you to create models of cells, just as you can, people tried through equations and algorithms to get, computers to be able to discriminate, cats from dogs in photographs. That failed over a period of decades. AI came along, and again, the ability of the AI to detect patterns if you feed it with right information, millions of pictures of cats and dogs with the appropriate kinds of reinforcement learning, enables the AI to learn in ways that we don't know what's happening in the guts of the machine, although we can program the machine to do this, and therefore to provide a model that is very useful in discriminating cats from dogs. In the same way, we believe that AI will enable us to create models of cells. And it isn't just cells. If you step back, the biggest success so far of AI in the biosciences, of course, is AlphaFold, rewarded with the Nobel Prize a little over a year ago. And the AlphaFold trained on the protein data bank. Leading up to AlphaFold, of course, in the 90s and the 2000s, was very strong attempts by scientists to use biophysical equations. In those cases, there are equations that you can have for protein folding, but people couldn't figure out how to apply those equations to get beyond about a 40% or 50% success rate in predicting folding. AI comes along and it does the task. When trained on the right kinds of data and the right amounts, suddenly it can give you a 95%, 98% success rate in predicting folding. So that's the, going from artisanal to engineering. Again, in the case of protein folding, it's actually a biophysical problem. When it comes to cells and cell behaviors and human disease, again, there are no equations for that. But AI is an incredibly powerful tool to find those connections, to find those patterns, and thereby enabling us to model the behavior of cells, of patients, and advance knowledge that way.
Xaira made a big splash in 2024 when it emerged from stealth with a group of top-flight venture capital firms and $1 billion in committed capital. Bob Nelson of Arch Venture Partners and Vic Bajaj at Foresight Labs co-founded the company. You'd been president at Stanford University and stepped down prior to co-founding Xaira, where you're now chairman and CEO. You've moved between academia and biotech. How did all of this come about, and when did you see Xaira as your future?
Well, as you say, I have moved back and forth between academia and biotech. I started out as an academic at UCSF and Stanford, I'm a neuroscientist, focusing on brain development and brain repair. But then I decided in the early 2000s to move to the private sector, to Genentech. I was very blessed to be at Genentech during an extraordinary time. I was there for about 8 years, ended up as the chief scientific officer, and actually thought that my initial plan had been to move there for good, but then a great opportunity came to return to assume leadership positions at Rockefeller University and at Stanford. But I always remained involved with the biotech sector, being involved on the boards of small companies and some larger companies. I co-founded a company in that period. So I always had a front row seat to some of the greatest innovation on the boards of companies like Regeneron and Pfizer, and co-founded Denali. But nothing that I've seen compares to the potential that I believe AI has to transform drug discovery and development. So that was sort of my personal journey that made me primed, I guess, for what happened next, which is that Bob Nelson, and this is a little bit of the genesis of the company, Bob Nelson from Arch and Vic Bajaj from Foresight had gotten together and believed that the time was ripe to build a company that could tackle the application of AI to drug discovery and development end to end. Independently, David Baker, who of course shared the Nobel Prize along with the DeepMind group with Demis Hassabis and John Jumper, they had got the Nobel Prize for AlphaFold. He got it for using generative AI to design entirely new proteins, the first completely de novo proteins that have been generated using AI in this way. And David had decided with some of his colleagues, including our co-founder, Hetu Camasetti, who's our chief technology officer, that the time was ripe to start a company around molecular design. The two groups came together and realized that it made more sense for them to pool their forces. And that's how the three of them came together. And then they approached me about being the founding CEO. I've already described how I was primed for this moment. In fact, I'd been involved with Bob Nelson in previous companies. On the Agios board, he brought me on the Juno board. He was really instrumental in pushing me and two other co-founders to found Denali. So I'd worked closely with Bob over the years, and of course he had been sharing his ideas and Vic's ideas. And then when David came together, so I was aware of what was happening, and things just one thing led to another organically, and it just seemed to me this is an extraordinary moment with the power of AI that we can deploy to this problem of drug discovery and development that I'm so passionate about, with extraordinary founders and great investors and great founding scientists. And so that's what prompted me to take the role as CEO.
There are a lot of emerging tech bio companies that appear to be trying to do what Xaira is trying to do, although they may be more modest in their scale and ambition. To what extent do Baker's AI frameworks provide a significant point of technological differentiation for you?
David Baker's models, which of course he has published and made available in all open source, are a great starting point for the entire field. And we, of course, had a jump start by being able to access those models early. Our co-founders, many of them come from David's lab, including the first authors on the very famous RF diffusion and RF antibody papers, which made such a splash in 2023 and 2024. It's hard to remember that this area of de novo protein design using AI is only about 2 years old. So we have both David's involvement as a founder and scientific advisor. We have extraordinary talent from his lab in the company, including several of our co-founders. And so the differentiation, of course, comes from other things because others have access to what's been published. The differentiation comes from the talent on the one hand. We made a lot of progress with our algorithms compared to what's in the public domain. It's been published, and also data. So we believe that data generation, which we are doing at scale at Xaira, is a very important part of the differentiation of the company.
You talk about data generation, the term garbage in, garbage out, it long predates AI, but it's become a fundamental reality. AI is only as good as the data which it's trained on. What's the range of data that Xaira is using, and is there anything unique there?
Yes, there's some unique things, and the data has to be adapted to the problem at hand. So in the case of molecular design, we're focusing currently on designing large molecules. That's our major focus, the primary focus on antibodies, because that's where the technology is most advanced. And again, we and everyone has access to data that are in the public domain, particularly in the Protein Data Bank and also in some other sources as well. And that's what's enabled a lot of progress by many groups, academic and industrial, and of course our company as well, in the initial stages of developing powerful models. At the same time, it's very clear from our own experiments and our own work inside that to really get to the hardest targets, which is what we're going after, that it's valuable to generate additional data. And we've been learning what kinds of data really move the needle. So we are focused on data generation. One has to be strategic here about what kinds of data to generate, and that provides sort of a unique differentiation of our company compared to others. I'm sure there are other companies that will, or maybe even some have started to generate data as well. But this is an area where those kinds of efforts will really enable progress into areas that are not currently accessible with just the data that are available publicly. Now, that's molecular design. We are focused also on this problem of target identification. And the approach that we've taken there is our aspiration really is to be able to build a causal model of the cell. First the cell, then tissues, and the entire organism ultimately, but it starts with cells. To build a causal model of the cell, we also believe and the data really support that you need causal data. There are a lot of descriptive data out there, a lot of terrific, for example, single-cell RNA-seq data. Tens of millions of cells have been sequenced, publicly available, beautifully collated by the Chan-Zuckerberg initiative, for example, and other databases. Models that are trained on descriptive data turn out to be very good at descriptive tasks, and they pretty much fail entirely at causal tasks, which is what we're interested in. For example, if you have a cell that's in a disease state and you want to bring it back to a healthy state, what you'd like to know is what genetic changes should I make in the cell to affect that transition? Well, today, the powerful technologies that have been deployed in a static way can tell you, there are so many genes, let's say 300, that are different between the diseased state and the healthy state. But if you ask the model to predict which genetic changes would take you from one to the other, the success rate is no better than a coin toss currently. So those models fail. The reason at the root why those models fail is because the model doesn't know what it doesn't know. It's never seen causal data, and so it fails at causal tasks. So that's prompted us, as well as some others, to generate causal data. And so this is the other kind of data that we're generating at scale currently, Danny, which is to very large scale, genome scale perturbations of cells. The workhorse technology that we're using, it's not the only approach, but certainly the central one we're using is Perturb-seq, developed by Jonathan Weissman and colleagues at MIT, which we've industrialized, where you can, in cells, modify one at a time each gene in the genome, and then do a very large scale readout, all the effects on all genes. So it's a 20,000 gene perturbation by 20,000 gene readout. It's even better than that because we've been able to now do partial gene knockdown. So each gene is not fully knocked down. Rather, we have a range of knockdowns and a range of readouts. And it's those kinds of data sets, 20,000 genes by 20,000 genes in multiple cell types, that provide the fodder, the substrate for generating a model that can start to understand cells at a causal level. Back in June, we actually published the method that we'd developed so that others can replicate it, which has much better signal-to-noise ratio, fewer batch effects than previously. And we also, in the process, published 2 data sets for 2 cell lines that we'd use to pilot the approach. There's been a huge interest in those data sets. It's the largest Perturb-seq data sets ever published. We've made them freely available for academic use and also for commercial licensing. There have been over 80,000 downloads of the data since we posted them in June. It shows you the kind of hunger there is in the field for these data sets. And that's just the beginning. Now we're at the end of the calendar year. We've generated 3 times as much data in the intervening months, not just in cell lines now, but also in primary cells, some of it in vivo, and we're going to be accelerating that further in 2026. We believe those kinds of data are necessary to provide the substrate to create the first causal models of the cell. Of course, with AI, you generate a model with data that are more readily scalable, like the kinds of data we can generate in vitro using Perturb-seq. But to where you really want to go, where to go for the gold, we want to focus on cells that are therapeutically relevant and in particular, some data that's harder to come by. So if you have a first foundation model, you can then fine tune it on data that's harder to come by. For example, data from diseased patients and healthy patients to teach the model how to discriminate that. And then through this process of transfer learning in AI, the expectation is that the AI will be able to provide insights such as how do you get from this disease state to this healthy state. So it's a program of data generation to create a model that will be understand biology at a causal level and enable us to make predictions that are therapeutically relevant. That's for cells. Ultimately, we want to tie that, of course, to patient data. And there you need other kinds of models and other kinds of data as well to represent patients and then tie all of it together. So a lot of data generation and different kinds of data for the different circumstances for molecular design, for target ID, for patient stratification, and then pulling it all together. The beauty of AI, of course, is it learns, so you can go through cycles, and if your predictions aren't great, you can feed that back to the model and figure out how to improve it. And AI also is uniquely good at dealing with multimodal data, so you can pull data together from all of these different sources. So that's the aspiration, if you will. We're doing the systematic work to do all of that. We're also being very, very pragmatic about applying this to use cases that can generate value in the near term in terms of generating therapeutics and insights.
And to go back to the synthetic biology question, are you generating novel proteins that don't exist in nature, or are you able to engineer intelligence into an antibody?
Depending on what you mean by intelligence, of course, specific properties, we can certainly infuse that into antibodies. And we are making a lot of proteins that don't exist in nature. In the past year, so we have our model for antibody design, which we call X design. All of our models start with an X for Xaira. So X design, X cell is for the virtual cell. X design. Our models will we go in 3 steps. The model will dream up structures. The prompt will be make an antibody to this particular epitope on this particular protein. The model will dream up various solutions. We then take those imaginations, if you will, and filter them through a model we call X-filter that identifies the ones that are computationally we believe most likely to be successful. And then we make them in the wet lab and test them to see whether whether the predictions were correct. And some are correct and some aren't. And we feed the information back to the model to improve it. Over the past year, we have computationally evaluated about 1 billion designs. We have selected about 1 million designs that we've actually made and tested in the wet lab and fed the information, the successes, the failures back to the model to improve it. So we are generating data at scale, and yes, we are generating proteins that don't exist in nature with the purpose of improving our models.
And how are you thinking about the construction of a pipeline and the indications and modalities you'll pursue?
Well, in principle, and this would be very specifically for X design on the design of antibodies. In principle, you could apply this to any targets. Of course, there are lots of great methods for making antibodies already through immunization, say of humanized mice, or B-cell screening, or yeast or phage displays, and others. And many of them have been very nicely industrialized, and they work very well for a lot of targets. Think if you have a single pass transmembrane protein, you just want to make a binder to the ectodomain. You don't need AI to do that. You could use AI to do that, but you don't need AI to do that. And currently the AI is still progressing. However, there are many targets, many high-value targets that are not well served by current methods. Think multi-pass membrane proteins with just a small amount of real estate, if you will, on the outside of the cell, ion channels, GPCRs, tetraspanins. Think other use cases like agonist antibodies, especially, say, to heteromeric receptors, where the antibody has to bring the 2 proteins together in just the right ways to activate the receptor. None of those are completely impossible with current methods, but they often fail. And people, might try, fail, and move on to other targets. So there are a number of targets that are have some or a lot of validation, but have been poorly served by current methods. And that's a place where we believe that AI can have a more unique impact, and which enables us to differentiate as a company, of course, and in a very competitive field. And in biotech, it goes without saying you really have to be able to differentiate from what others are doing. So both because it will showcase the potential of the technology best, and because it enables to differentiate, we are squarely focused currently on targets with some or a lot of validation, but have been considered undruggable by current means. So that means we're not focused on particular therapeutic areas or indications. Rather, we're focused on targets that meet our criteria. So, in fact, we're active in multiple disease areas currently, and we plan to move a portfolio we're moving a portfolio of therapeutics forward in multiple areas.
How do you address the issue of manufacturability? Are you applying AI to determine that as you identify potential candidates?
Absolutely, and that's a great question, Danny. The AI is beautifully adapted to this problem. So when we get a hit, and if you got a hit by other means, for example, with a phage screen or e-screen, you typically have to go through a number of steps to go from hit to lead to optimized lead to development candidate. You want to make sure that your antibody, in our case, has the appropriate properties in terms of solubility and thermal stability. You don't want it to aggregate. You want it to have very low polyreactivity, high affinity, high specificity, low immunogenicity. All of those are properties that can be, especially all of them up to immunogenicity, can be modeled very straightforwardly, where you can generate data, and machine learning can be applied to learn what kinds of sequences and structures will give you the best shot at the best properties. So we are doing that as well. There are some data out there already. There are models out there that can be mined. We are generating a lot more data to teach our AI those properties. And this gets back to the point we were discussing earlier. I mean, currently we are going for these undruggable targets. As I like to say, we're not currently going for the low-hanging fruit. We're going for the high-hanging fruit. And to go for the undruggable targets. And what we're getting out of our models currently are hits that we then have to improve for a combination of AI and other methods to make optimized leads, development candidates. As we generate the data, as we go through multiple design, make, test cycles, we are teaching the AI all those properties. And so the aspiration is that in the not too distant future, rather than just getting hits out of our model initially, that we'll get something closer to leads, or maybe even optimized leads, or even a development candidate. So we believe that over time, our models will provide us with a time advantage as well in terms of making antibodies. We're not there yet, but we're pushing hard to get there as soon as possible. But again, where we're focused primarily today is to have powerful enough AI to go after the high-hanging fruit, so that we can go for these undruggable targets.
And when you go after the undruggable targets, is the AI identifying alternative binding sites that you're able to hit?
We definitely, the AI assists us in evaluating the binding sites. Sometimes you want to define the binding sites through other methods, perhaps empirical. For example, if you want to have an agonist antibody, identifying, doing an epitope scan or something like that. But the AI, it's a combination of AI and wet lab work that helps us identify which are the best epitopes to train our AI at to make the antibodies.
Xaira's building its own pipeline, but how are you thinking about partnerships and the role they'll play as you grow as a company? Or is that something you expect to pursue?
The short answer is yes, absolutely. The way we've looked at it, you might ask, why are we building a pipeline? There are many companies out there that are setting themselves up as platform companies to do one or other of the things that we're focused on, whether it's virtual cell bio AI models or molecular design. From the get-go, we thought uh, uh, we wanted to tackle a problem, both building the platform to, remember, our aspiration is to help transform how we discover and develop drugs. So we want to build a platform that can help with that. But the best measure of success and the best test of our models is to show that it will enable us to generate a pipeline of differentiated therapeutics that can make a difference for patients. And so that's part of the reason we capitalized the company the way that we did, so that we could pursue building the platform and building a pipeline. And we wanted to be able to do it without having to go back to the capital markets until we'd shown that the success of the platform, and as measured by having multiple products in the clinic. So it certainly is our base case is to develop our own therapeutics, but we're also very open-minded because it can often make a lot of sense to partner. So in case of some therapeutics, we may want to partner because the partner has specific scientific capability or clinical capability in particular indications. It may be desirable to amortize risk by risk sharing with a partner. So for every one of our therapeutics, we'll be asking, is it something that we could go solo? And even if we could go solo, do we want to go solo, or do we rather partner it? But it'll be driven by pragmatic considerations like that. So that's in terms of our pipeline. We're also, we believe that we could go further faster if we were to partner with, a small group, or perhaps even just one major partner, to advance the technology simultaneously as we build a pipeline and do it together with them and with all the resources that a partner can bring, including data and expertise. So we're very open to the idea of partnerships, but we also have the wherewithal to go it alone if we so desire.
One of the advantages you have is that you're not bringing AI into an existing operation, trying to get people who are used to working in one way to now integrate AI into the process. How are you thinking about talent in terms of tech versus biotech? And is there now a large enough base of people with experience in AI-driven drug discovery and development to hire people with that background?
Yeah, that's a really great question, Danny. And the short answer to the second question is no. There isn't a broad enough base of people who are bilingual, if you will, in both AI and biology. If you look at the people that we brought together, so we have just extraordinary talent, AI scientists. We have disease biologists and therapeutic area experts. We have seasoned drug hunters who have taken hits and turn them into drugs, and clinical trial specialists also, so moving them through the clinic. Each of these people is an expert in their area, and most of them not only are not expert in the other areas, but in many cases don't have great and deep knowledge at the other. But what they have is, first, the excitement of coming together around this mission, and the excitement of working with people who have that expertise. So in fact, my role as a CEO is first and foremost to identify the talent and bring people together and to have them working together productively, even if they have non-overlapping skill sets. I can tell you, we organize our scientists around programs, therapeutic programs, and just the level of excitement when the AI scientists learn about the biology of an area and what it takes to make a drug, when the biologists see what AI is capable of, when the drug hunters learn from both of those sets of people, it just creates enormous enthusiasm, brings people together, and bit by bit, each of these people is becoming bilingual or multilingual. I think with this generation of people, that includes myself, what you have are people who started with one language are learning another. You're an AI scientist, you're learning the language of biology and drug discovery, or vice versa. And you might say, if you'll permit the analogy, that all of us will grow up and will, like people who learn a second language in their 20s or 30s, we'll have an accent when we speak the second language. there's a younger generation of scientists, some even in universities right now, certainly some of the younger scientists who are joining us, they're learning the other language at a young age, and they will be bilingual without an accent, I predict. So 10 years from now, you'll have, I think, a whole cadre of multilingual individuals from companies like ours and academia as well. But right now, what we're doing is putting together people who speak different languages, but who are just so passionate about the mission of making medicines that can help patients, and excited to learn from one another.
At the end of the day, what do you think Xaira will look like culturally relative to a more traditional biopharma company?
Well, culturally, we're an AI first company, and we want all of our scientists to be AI first, and we want them to acquire this bilingualism or multilingualism as well. So culturally, what you'll find is, and we are building the tools. I haven't talked about the fact we're also building AI agents to enable all of our scientists, for example, the AI scientists who want to learn about the biology, rather than having to ask a colleague, they can have the agent help them, vice versa. Our biologists, when we started, if they wanted to look at a particular design model or structure, they typically call up a colleague who is a structure, AI structure specialist. And we're building agents so that they can do a lot of that work on their own. This won't stop them from talking to the others, but it means that they aren't always having to pause and find someone who has the time to explain. Rather, they can work through it themselves before they talk to the specialist as well. So what we want are people who will be fully fluent in these areas and able to use AI agents to advance their science much more effectively and more rapidly and more deeply, even as we maintain the cohesiveness of this team. And I think that will be it's certainly different from how biopharma companies are organized today. Over time, I think any biopharma company worth its salt will want to transition to that. It's just that we're set up to do that from the get-go, so we'll be in the vanguard of that. But of course, it's important for all of the industry to go in that direction. That's certainly something that we'd like to help catalyze as well.
When I think back to sort of the first generation of genomic driven drug developers, the lesson there was they were all going to build these massive pipelines very quickly, but the rate limiting step became the capital needed to do clinical trials, particularly later stage clinical trials. And I'm wondering if we're going to see the same thing with AI. You've done a massive fundraise for a startup, but as you advance candidates into later stage clinical trials, it becomes very costly. What does success look like, and how large a pipeline do you think you'll be able to support?
Well, we obviously would love to be as rapidly as possible in the situation that you're describing, that we have so many candidates that are ready for clinical trials that we have to deal with that issue. And so we want to get there fast. As we progress and we get those candidates, I think you said, it's about getting better molecules faster and more cost effectively. Remember, today we don't have that speed advantage yet with AI molecular design, although I think it will come in the near future. As we develop molecules and line them up and have them ready for clinical trials, obviously there are many possibilities. That would be an embarrassment of riches scenario, if you will. And you're absolutely right. The $1 billion we raised is a lot of capital for a startup. Enables us to really pursue, both our platform strategy and our pipeline strategy, and to go at it in a deep way with a lot of security that we can keep moving and improving. But as we get to that point, as we generate value, of course, there will be opportunities either to partner or to raise additional capital as we go along. So it would be a high-class problem, if you will, and one we would welcome very much, and one that we think we can navigate as well. Stay tuned. But right now, we're mostly focused on the first part, which is to develop that technology to make better molecules faster. And I should say, again, right now, what we're trying to make are molecules against targets that have been difficult to access, the so-called undruggable targets first. But over time, we believe we'll be able to make better molecules faster and more cost effectively with AI. And when we get to that point, I'm pretty sure we'll be able to solve that equation.
Marc Tessier Lavigne, co-founder, chairman, and CEO of Xaira. Marc, thanks so much for your time today.
Thank you very much, Danny. I really appreciate you having me on the show.
Thanks for listening. The Bio Report is a production of the Lavigne Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to lavignemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@lavignemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.